Dr. Eduardo Jule. Dr. Eduardo Jule earned his PhD in materials engineering from University of Tokyo, where he specialized in designing polymer-based nanoparticles as cytotoxic payload carriers. He later transitioned to a business development role at Nanocarrier. Following that, he joined Capsugel and spent 13 years focusing on development and manufacturing services for small molecules in various capacities, including head of formulation and product development and specializing in lipid-based drug delivery systems. After serving as regional head of sales at Lonza for 5 years, Eduardo left the company in 2022 and established Evolve Consulting and has been a consulting there since then. So Eduardo is going to talk again. We hear several times today, lipid-based formulations. They are not gone. We still have lots of formulations that are being developed using lipid-based formulations. So let's learn more about that from Eduardo. Thank you, Eduardo.
All right, good afternoon, everyone. Today's talk will be based on product performance optimization. And I'll give you a heads up on what the key takeaways are. Product optimization can be achieved sometimes by simple formulation optimization, tweaks in the formulation that leads to better performance. And most importantly, they can be achieved simply by choosing or matching the right enabling platform to a problem statement. And of course, I'll be talking about lipid-based because that's what I did in a past life. So what does it matter, right? Why selecting the right technology matters more than ever. I'm not going to bore you with the details on how the overwhelming majority of new chemical entities hailing out of drug discovery. We've heard about this, right, a number of times earlier today, which is good. I don't have to repeat what's already been said. But the essence of the challenge here is that, of course, we have limited solubility, which leads to poor and/or erratic bioavailability. And as if that was not enough, the challenge is that formulation scientists now need to deal with their assets in a way that you deal with a portfolio. That is, you make sure you need to make sure that you progress the promising candidates from a formulation development perspective as fast as you can, and then you kill the ones that aren't even faster, right? And on top of that, so the pressure is there. They're often prey to, or victims, I call them, of the trial and error approach, whereby they try sequentially different platforms, different techniques and technologies, right, in increasing order of complexity, which doesn't work well for lipids because they tend to be what's left when nothing else matters, nothing else works, right? Sometimes they try different things in parallel, but also oftentimes they're victim to the what you know or who you know, right? What technology you have experience with, what vendor, what salesperson you like, and I like that guy, so I'm going to give it to them, right? I'm going to end up working with the same, again, same technology, the same CDMO, just because that's what you're used to. And I'm not being critical, I'm not here to judge anybody. I'm just saying that this happens, right? Not to everyone, but even to the best of us. We've been victims of it. The great news is that, see, when I started working with drug delivery systems back in the early 2000s, with nanoparticle-based drug delivery, more specifically, I knocked on pharma's doors and they would tell me, well, we know how to make tablets. Well, what do you mean drug delivery? We come back when you're in phase 2, phase 3. It was perceived as a sort of a science fiction thing. But in the meantime, of course, it's become commoditized, and we continue to see a number of exciting new technologies. My simple graph here is obviously not complete. There's new, novel, and exciting technologies that continue to come to the fore, which is great because that gives formulation scientists the choice. But sometimes the choice it feels comforting on the one hand, but on the other hand, making sure that you're, again, matching the right enabling technology to a problem statement. There's no one-size-fits-all type of approach is absolutely key because it can have an impact on performance. And if there's anyone from Janssen here in the audience, I'm not picking on Zytiga. I'm just using a case study to basically make a point here. At the time of launch, 2011, I believe it was, Zytiga or abiraterone acetate was approved and quickly became a standard of care for the treatment of prostate cancer, continues to be a standard of care. But when it was launched, the product came with a label clearly stating, do not take this with food. And now, mind you, of course, food effects are not uncommon. But in the case of Zytiga, the jump in Cmax and in AUC was over 10x, 17x and 10x respectively. So it was a massive food jump, which obviously was a concern to the point that was clearly advertised as do not take with food. 7 years later, Sun Pharma launches Yonza. See the difference, 125 milligrams in tablets. And the product was approved, and it features a couple of interesting things, key and very tangible patient benefits, at least from a patient's perspective, which is obviously important here. First, no food restrictions, which obviously gives the flexibility to take with or without food. Increased absorption, because we now go from 4 tablets at 125 mg or a 500 milligram dose versus a 1 gram dose previously. And this is based on studies that demonstrate bioequivalence, right? Similar bioavailability to that of Zytiga, but now at a lower dose. Now, what's the secret here, right? What was the magical enabling platform they used? Nothing more but solid matrix. And I'm glad Jason talked about that before, because that checks the box, the topic's covered. But what Sun Pharma did here was basically reducing particle size from the micron range to the nanometer range, and that basically increased, we've seen it, right, absorption, surface area, of course, which leads to increased dissolution rates and enhanced absorption. Put another way, a simple formulation optimization, because again, there's nothing fundamentally revolutionary about the approach, but optimizing an existing product, right, leads to substantial improvement in product performance. And in this case, again, very tangible benefits for patients. And I use this, again, not to talk about, again, Zytiga is an undisputed success story, 3.5 billion peak sales, and combined 10 billion in 2019 to 22, and after losing exclusivity. So it's a great success story. But if we think about it holistically from a product development perspective, right, I usually invite the audience to think about what, developing a suboptimal product can mean for your own drug, of course, and for your portfolio more broadly, and to the patients you're trying to serve. So I'm not going to, again, because many of these things have been talked about, but just to briefly touch on understanding the different techniques and technologies available today. We know that molecular dispersion continues to be, and that's one of the key things, right? The key requisites to increase solubilization, bioavailability, still goes through the active itself, understanding first what it takes to drive absorption, and solubilization, of course, can be limited by solid state properties. For brick dust, the behavior will be different here, or solvation for grease balls. It's compounded by dissolution rate. And again, I'm not going to talk about this, but then there's multiple techniques and technologies available today. There's top-down particle size reduction. We've heard extensively about micronization. Nanomilling, for example, or bottom-up nanocrystallization can, at the end of the day, lead to, again, increases in surface areas, right, which can lead to dissolution rates. Some of the limitations here is that even if you achieve immediate dissolution rates, you're creating high energy states that need to be stabilized. Less, of course, you go back to, you have reaggregation, you have introduction, undesirable introduction of amorphous materials, for example, which is why solid dispersions have become a golden standard when it comes to bioavailability enhancement. It makes a lot of sense. You're now dispersing very often at the molecular level and onto a high molecular weight polymer matrix, which is great, right? You have now increasing surface area to facilitate hydration and dissolve the API now ideally from an amorphous state. This is beautiful. Continues to be some of the initial concerns we had about stability. All of those have been addressed by smart engineering. Spray drying especially has become a standard, golden standard today, which is great, right? And we know that all of these has an impact on, leads to increased solubility and dissolution rates. And again, particle engineering is one of the golden standards. But again, there's no one-size-fits-all type of approach, right? We heard it this morning in a very vivid manner. We tend to fall back on what feels comfortable, what we've done before, and this can sometimes be a mistake. But okay, going back in time now a little bit, talking about another case study where a simple formulation optimization process led to substantial product improvement or product performance improvements. So this is at a time when clinicians had a very hard time dosing cyclosporine. Sandimmune was launched by Sandoz in 81, then optimized into Neoral in 95. See that both are soft gel capsules, right? But there's a fundamental difference. Again, the challenge that clinicians had was that the absorption profile they saw in patients was extremely variable in the presence of food. They saw variability inter-patient, but even intra-patient, and that was a challenge at the time of dosing what's in effect an immunosuppressant. Neoral, of course, a self-microemulsifying, so we went from a self-emulsifying, so a very coarse emulsion, to a very fine microemulsion. Evidenced in the clinic the three key benefits of these type of formulations. And again, I'll be talking about LBFs a little bit more in detail. But obviously, one is the bioavailability enhancement, the reduction of food effects, and the reduction of variability that weren't properly understood back in the 90s. And one of the main reasons, and I'll revisit some of the reasons why lipid-based has been challenged in the past, but also we'll talk about why it makes sense to revisit today more than ever. But again, the takeaway from these two case studies, right? In the case of both Yonza and Neoral, small formulation optimization changes led to substantial changes of major impact on product performance and with very clearly tangible and very clearly defined benefits for patients. So when I say that we have come a long way, we know that in the case of grease balls, right, some of the solid state limitations can be bypassed by complexation, and by complexation I mean, for example, inclusion in cyclodextrins, or in the case of more lipophilic type of APIs, by solvation or solubilization in lipophilic environments. Now, of course, not every single API out there has a log P over 5, but it's been demonstrated that that's not the only type of property that makes an API amenable to this type of formulation, just like not every single excipient out there is an oil, right? A lipidic vehicle. We work a lot with, I say we as in my past life in product development, but we work extensively with lipids, oils, semi-solids, surfactants, co-solvents. We heard it earlier in the GLAD talk as well. And there's a great paper in 2007, where Chris Porter very elegantly elucidated three key mechanisms of how lipids work, what sort of an impact they have. First off is by enhancing drug solubilization, so we now have solvation of the API into whether it's an oily vehicle, a self-emulsifying, microemulsifying type of formulation by formation of vesicles, of course. That's what we're trying to achieve, to disperse the API in a vesicle mixed micelles as well, because as we will see, lipids are unique in the sense that they are digested, of course, and this introduces another variable into the equation. This potential interaction with enterocytes, for example. We talked about, we heard talks about the potential inhibition of receptor-mediated efflux, for example, pre-systemic metabolism, which has been demonstrated in vivo, even not necessarily in humans, but there's clear evidence that they may have certain excipients can have an impact at this level as well. And third, I'll briefly talk about this later, the possibility of altering the drug absorption pathway, redirecting from the portal vein into the lymphatic route, which is another attractive way of bypassing first-pass metabolism. There are products in the market that claim this can be done, right? And there's sufficient evidence in animals and in humans that this can be achieved. So that's an interesting benefit. But for the sake of this conversation, I'll focus primarily on the first point. And as I said before, uniquely for this type of excipients, not all of them, but still, even small amounts, 1 to 2 grams can trigger, if not full, drug absorption patterns that are similar to those observed in postprandial administration. In other words, lipids are recognized as food. If they're recognized as food, they are digested. As simple as that. And the reason I put the crappy graph there is to show the behavior of different excipients just by themselves on digestion. See that some excipients will be digested, and what we measure here is the amount of free fatty acids being formed as the excipient is degraded. Of course, some of them are not. And the key question here is, does it matter? Well, obviously it matters. What's best? It boils down to the API. How does the API behave in that formulation, and how does it behave on dispersion digestible? I'm getting ahead of myself. So as they are digested, of course, lipids move down, and I'll skip all the details on how that works, but as they move from the stomach, the gastric environment where digestion begins, into the duodenum, you have a funny combination. You have undigested materials, because only about 25% that get digested in the stomach. You have undigested materials, you have colloids, you have mixed myocyls now, you have a combination of exogenous and endogenous materials coming together. And in this very complex, evolving and dynamic environment, what we care about is, where's the API? What happens to it, right? Ideally, of course, the API stays in solution as the formulation around it is or isn't digested. In some cases, we have shown that it's possible to supersaturate the active. That is, the compound actually benefits from those digested phases and sees its solubilization increased to levels that wouldn't be otherwise, that couldn't be achieved otherwise. And this is when it gets really exciting, because what drives absorption, we know, is the creation of a concentration gradient, right? You need to have a gradient that pushes the API through the unstirred layer. We heard about this earlier today as well. See, it's great. I can skip a number of things because they've been talked about. And then, of course, once you achieve that supersaturation state, there's a certain threshold over which, of course, this can be counter-effective. Now the API starts crashing out, and this is, so finding the right balance. But at the end of the day, to begin with, understand where the API is, right? Does it stay in solution? Is it supersaturating? Or, of course, is it precipitating? It could also be that the API is no longer soluble in digested phases and then crashes out. And this is why, in the past, these type of formulations were a little bit looked down on because they needed to be tested in animals, right? There was over-reliance of animal models. We developed great formulations. Hey, it solubilized the API, but this is all great. Put it in animals, doesn't work. And why? We didn't know. We didn't know what was affecting the performance. But take away from this slide and from the previous points is that we have here a technology that can harness naturally occurring phenomena, whether it's digestion or lymphatic absorption, right? It's unique in that sense. But what's key, of course, is understanding, right? Controlling and ideally, to some extent, predicting how a system will behave, what is going to give us the peace of mind, the confidence that these formulations can actually be developed and considered like any other enabling platform. And this is work that was done back in the early 2010s. Colin Powden, Professor Powden out of Monash University, proposed the LFCS, or the Lipid Formulation Classification System, initially as a means of describing the different types of formulation by classifying them into different types. And the types were based on the composition of the formulation, right? There were fully oily vehicles to non-oil SMEDs, self-microemulsifying type of formulations. But then he realized, but this is not enough, right? The composition is not what matters. What matters is how they behave. And then he came to identify 2 key performance criteria. One of them was, already mentioned, digestion, but before that there was dispersion. And I was laughing, again, I think many of us were this morning on hearing that about dissolution, right? Many times I've had conversations with clients evaluating a SEDS or a SMEDS who tell me that, they run dissolution studies. And I'm like, but the active is in solution in the sets and that it's dispersed. Well, why are you running this solution? No, it passes 100% of the times. What's not to like? It's great, looks great, the data looks great, regulatory loves it, so we keep on doing it, right? But importantly here is that when we created the consortium, the LFCS consortium, the idea was to harmonize how things were looked into. And we put together a number of people from universities, from the industry. Long story short, the consortium came up with harmonized directives and protocols on how to evaluate dispersion and digestion. So these guidelines continue to be used today and give us the ability, and we've seen in multiple case studies, the ability to predict vivo performance based on vitro testing. And this is beautiful now, because now we have the ability to some extent to understand and to predict performance based on simple vitro studies. Sorry about laser pointing there. And this also has helped us understand what happens to Sanimune in the oral. I'm not gonna suffice it to say that in the case of Neoral, we have undispersion, right? Remember I said Sanimune was a coarse emulsion. Neoral was a nice, fine microemulsion. On dispersion, you have a more consistent digestion profile because you have a very nice and monodispersed particle size distribution in the case of the microemulsion, which is what you see on the right side. Sorry, that's the oral in the FASIV and fed. See that to begin with, most of the API, which can be found, sorry, it's easier if I get closer, the API can be found in non-digested, oily, and aqueous environments, which is where we want it to be at the end of the day. On dispersion, Neoral already has all of the API in solution, which is beautiful, and on digestion, that API stays in solution, which is what we want. In the case of Sanimune, of course, it's a coarse emulsion, so it's poorly dispersed. It benefits from the presence of food to be absorbed, right? It gets hijacked along with the food, but see how the performance is extremely variable. And what matters is the concentration of free drug at the end of the day. When you look at the difference between the two upon dispersion and digestion, to begin with, the concentration of free drug in the oral is already higher to begin with. Drops a little bit on digestion, but it stays the course basically, and it's higher than with Sanimune. Forgot I had the mic here, I could walk around. But anyway, back to abiraterone. Conscious of time? Simon, no scoreboard? Okay, super. Again, product optimization was achieved by reducing particle size, which is great. We compiled some of the data here, right, from the use of micronization to milling by solar matrix. See that bioavailability was increased from 20 to 25%, reduction of the dose, reduction of the food effect, and of course, that got you a nice label here, or the absence of a label in this case, of course. But as I said before, no one size fits all. So the question here was, okay, moving bioavailability from 10% to 25% was good, right? It's a great achievement, but could it be better? And the short answer is yes. And in this case, we used LBF because I remain convinced, and I was telling some folks earlier today, I'm not here to represent any CDMOs. I'm here as a former product development manager and a big fan of this type of technologies. Now more than ever, it's a great time to work with these type of formulations because we have come a long way. We understand the science. We have advanced the science to understand what drives absorption, what the key performance criteria are, right? And to some extent, we can predict performance as well. When we looked, and this is work that was done in a previous product development life, at the properties of abiraterone, because that's one constant, right? We still need to go through very careful, thorough evaluation of the property, the characteristics of the drug, of course, make sure that we find the right fit. It's not defaulting back on what we know or who we know, but look into the properties of abiraterone and see that it's obviously very poorly soluble, has low solubility, it has a high log P, a moderate melting point, and you heard this before, right? These makes it very amenable to either amorphous dispersion or LBFs, but what, from our perspective, made the difference is that Zytiga was subject to massive food effects. And the Summary of this is we followed exactly the same approach. We developed formulations that were tested on dispersion, that were tested on digestion to identify the lead formulation. And we took that lead formulation, we dosed it in dogs. We're using Zytiga as a benchmark. And in that study, so this is vivo data, we achieved bioequivalence. The interesting thing is we achieved bioequivalence at 50 milligrams instead of 250, so that's a 5x increase in bioavailability. Statistically, food effects were suppressed and variability was substantially reduced, right? So again, in some cases, simple formulation optimization, in this case, matching or better matching, more accurate matching of enabling technology with problem statements can lead to substantial, again, improvement on product performance. So I'll let you read this at leisure. I try to address some of the earlier concerns around this type of formulations. They're complex, they're expensive, they leak. Even today, we continue to hear this. Most of these concerns have been properly addressed. And I speak on behalf of many of the experts out there who have, again, advanced the understanding of how lipid-based excipients work. Talk to anybody at Gatfosé. The chemistry around it, which is essential, of course, when we do solubility screen at the pre-formulation level. It's not a matter of screening different excipient suppliers. It's about understanding the chemistry behind each and every excipient type, understanding what makes sense based on the problem statement, of course. Expensive, leaks, all of these to me have been observed. Now, this is not to say that they are the solution to every formulation challenge out there. Absolutely not. This is just to say that they are another very viable platform that's been around for decades, that it's easy to scale up, that's commercially accessible today. But that's it. I mean, in the past, we've seen that there's multiple reasons why drugs end up in a lipid-based formulation. In some cases, it's very straightforward. It's very simple. See, in the case of how do you go back? Here. In the case of dronabinol, Marinol, the drug is extremely soluble in lipid environments. It was easy to solubilize in sesame oil. In the case of tafamidis, if my intelligence serves me right, the active was a nightmare to deal in a dry form. It was sticky, it was a mess. That's why they ended up putting it in a soft gel. In the case of lubiprostone, the drug is not even absorbed. It's used for the treatment of IBS, so it's absorbed in the small intestine. But the reason it was put into medium-chain triglycerides is because of the low dose, to achieve content uniformity, to facilitate the analytical work, because the doses were initially 24, and then 8 micrograms. So it just made more sense to put it in a liquid vehicle as opposed to a tablet. So sometimes the reasons are, again, more practical. In some other cases, they follow precedents. Voclosporin is a distant cousin of cyclosporin, of course. So there was a precedent there. And as I mentioned earlier in the talk, testosterone undecanoate, because of the potentially to bypass first-pass metabolism by redirecting to the lymphatic absorption route. But many more drugs, I'm convinced, could be reformulated. I'm not talking about, of course, can be formulated from scratch, but could be revisited, right, through B2 approaches by either optimizing formulations or, again, revisiting some of these concepts to come up with better performing products. Conclusions and takeaways. I think I've said these over and over. The commercial piece, of course, which is very brief, just to say that I'll be hosting a master class on lipid-based formulations for those who are still interested in this technology in June. And that's it. The last slide is for you to read again at home, for you to get a sense of what Evolve does today. Technical advisory is one piece of it. But with that, I'll be happy to take any questions. Thanks for your attention.
Any questions for Eduardo before we hop right on?
So either you're convinced or confused.
Yes.
Great presentation. Just a general question I have, because I've used all the traditional solid excipients and making tablets and all those things.
Yes.
As far as the available amount of excipients that are compliant with pharmacopoeias and things like that, what is the selection as a formulator with lipids versus if you're using things like, I don't know, a cellulosic or a methacrylic polymer like that I deal with? What's the general range of options you have?
So it can be a major problem in certain parts of the world. Places like Japan, for example, we try to identify excipients that are, anywhere near the pharmacopoeia, and there's almost none. But for the rest of the world, I think there's a sufficient data today to substantiate that. Many of them are compendial by now. Maybe not the same amounts, but still, they're commercially available. They come with extensive data packages that range from technical data understanding, the TDS type of data sheets, into tox data that can support any concern there. So as of today, I haven't come across any major challenges that I would say, yeah, this is definitely a hurdle. Maybe you have a smaller choice, but still you have a choice nonetheless.
Thank you.
Hi, awesome presentation. I have a follow-up question. So just like in solid orals where talc, titanium dioxide, these are becoming like bigger nemesis, or colors, which is becoming a big issue now. For lipid-based formulations, do you see any such trend, TikTok trend, that is potentially going to be disruptive? For example, lipids that are based on palm oil, or maybe trans fats or something, which is, already a big conversation in foods.
I'm going to look at my excipient provider friends here. Not that I'm aware of. I haven't seen anything. As with any other part of the pharmaceutical industry, there's increasing concerns about sustainability, of course, the origin of raw materials, how they're sourced. Absolutely. Is it more so for lipids than any other technologies? No, I don't think so. So no, I'm not aware of anything that's overly, concerning, let alone dramatic so far. But I'll look at.
Thank you.
No, nothing that you're aware of either. Okay. All right.
Thanks.
All right, thank you. Any final questions for Eduardo before we move on? No, all right, thanks very much, Eduardo.
Thank you.